at nanotubes, this weird idea that the microbes can create a tunnel, and it's just cytoplasmic extension, essentially, that will allow for one bacterial cell to connect to another, and then exchange of material can occur. Here's a list of what they've discovered so far. And it seems like it can be used for nefarious purposes or good purposes. It depends on what's going on, but they have demonstrated that one bacteria can actually use it as a tool, as a weapon to kill a nearby bacteria and then extract all the nutrients for itself. So very targeted kind of predator-prey relationship. And then sometimes they've seen in experiments that it can be more like a cooperation where you can get nutrient sharing going on, or it could be like more of a donation of one to the other to keep the other alive. And so they're finding, this is all experimental now, okay? So they're doing this in the lab and they're demonstrating that it can happen. What really happens in real life, like, is it more competitive? Is it more cooperation? Is it even a mixed bag? They don't know for sure, of course, but they're certainly demonstrating that you can get this occurring in lab experiments. And so here, the first experiments that they did was, I think this is E. coli. And so they were just demonstrating that one cell of E. coli can build a nanotube to connect to another E. coli. And then they started asking, okay, well, can it be two different organisms that aren't related to each other? And so they did some mixed cultures in the lab, and they were able to find that, yes, it does occur. So here's Bacillus subtilis and E. coli. And you can even visually see the difference between these under this magnification. And they were able to demonstrate that there are nanotubes, even though they're unrelated. So again, this interspecies idea, similar, I mean, not exactly similar, but the same idea of interspecies we saw in the quorum sensing idea. Now they're showing it can happen with the use of nanotubes. And then they did some nice studies where they did green fluorescent protein, which is easy to pick up under a fluorescence microscope. And they just were using this again to demonstrate that there are things getting passed. In this case, it was the green fluorescent protein through the nanotubes. And so the B. subtilis was tagged and had the green fluorescent protein, and then E. coli might not be so easy to see right now in this light, but very faint dots of green you can see starting to show up in E. coli. So again, proving that you can get transfer. So here they did this interesting experiment where they were looking at antibiotic resistance, and could nanotubes be helping the bacteria gain resistance? And so we looked at how antibiotic resistance is often gained through conjugation, where the plasmid is transferred, or one strand of the plasmid is transferred, and that plasmid has a lot of resistance genes on it. And that's through the sex pilus. Well, now they're showing nanotubes can also seem to be the way of transferring antibiotic resistance. So let me, so they referred in this paper, they referred to it as non-hereditary inheritance of phenotypes. So what they mean is that there wasn't a plasmid transfer in this case. They were showing that there wasn't any DNA transferred, but the idea that there could be messenger RNA or proteins getting transferred was a strong possibility. And so this idea is being shown in the bottom image, where they're looking at different species or strains. And so P1 and P2 are two different bacteria. WT that you see here, that's wild type. Okay, and then P1 is a microbe that is resistant to this antibiotic, chloramphenicol. It was growing in the presence of chloramphenicol, but it got wiped out in the presence of lincomycin. So that's how you interpret what's going on here with, so the essentially this far left column is your control. LB that you see there. Let me blow this up into the next. This is a little bit bigger. Okay. So LB, that's just Luria broth. It's just, let's just grow them up under normal conditions, and you can see they're all growing without any antibiotic. Then they said, okay, let's take some, a flask and add in chloramphenicol. And when you do that, you can see that P1 is just fine. It's like, yeah, I can handle chloramphenicol. So it's resistant, but it got wiped out in the presence of lincomycin. Okay. Then if you look down at P2, P2 is able to grow in the presence of lincomycin, but can't grow in the presence of chloramphenicol. And then if you mix these two species together, so P1 plus P2, the purple option there, you can see now both P1 and P2 can grow in the presence of both antibiotics. So when they were investigating this, they were demonstrating it was through a nanotube process that you had this transfer of the phenotype of resistance to the antibiotics. So this is yet another way that antibiotic resistance can spread, which was a downer. Yes. I would think the sex pellets would be more expensive to make because it's protein subunits, whereas some membrane material is definitely more rapid to make. I would think it would be energetically less intense. So I think the nanotube might have an edge in that regard, that it's a little easier. But that's my guess. I'd have to see if I can find anything on that more concrete. So yeah, this was a study done. It was really interesting, but unfortunately the outcome was not such good news because now this is another way for antibiotic resistance.
So this is like proteins that are.
It could be proteins.
Okay.
And they didn't, so when they published this, they didn't get that deep yet.
Okay.
They were able to show it wasn't any kind of plasma transfer, but they said it could have been like a chunk of messenger RNA that codes for resistance that got transferred, or proteins that allowed for resistance that got transferred. So yeah, they're following up on that, on this study. This was nicely done, but they need to, there's still a lot of unknowns when it comes to nanotube processes. This is one of the newest discoveries of how microbes communicate and share things and information. They've even done some studies here. The title, Microbial Assault and Thievery via nanotubes. And so here they have two different colors showing the two different bacteria. And so B. subtilis is the really green one. And what they do is they have B. mega in red. And if you look in figure A, you see a ton of green and very little red. I'm sorry if you're colorblind, that's probably not telling you much, but most of what you see in figure A is all green and just a little bit of red. So basically, what they were showing in this experiment is that B. subtilis through nanotube processes was transferring toxin and killing off B. mega. And so you see that B. subtilis survived, very little mega is present. And then when you get a mutant, so then they said, well, let's take B. subtilis and knock out its ability to make that toxin. Then you can see that B. mega was much more capable of growing, even though B. subtilis is there. So that study was, again, showing toxin production and flow through the nanotube. And then they were showing that part of the process is, like, why is B. subtilis doing that? It's doing that to kill the bacteria and then essentially suck away the nutrients, so that it can have all the B. mega's nutrients to itself. And so it's a predatory relationship, really, in those conditions. So nanotubes, interesting work going on this, but yet another way that microbes are interacting in a social construct. Some good, some bad in terms of positive, negative. And now the last thing I wanted to talk about is extracellular vesicles. So these are not just microbes that can do this. Like mammals can do this, plants can do this. So it seems to be kind of a universal thing. But basically, it is, and some of this material is in chapter 3, just a little chunk of it. So it's in the Word document of assigned reading. You can find it in chapter 3. But this picture is showing some, I think this is E. coli with a bunch of extracellular vesicles that you can see along the body on the cell of E. coli. And when they started to look inside all of these vesicles, like what's going on here, they discovered that there's a range of different things in there. Just depends on how you manipulate the environment of the microbe. And so they found everything from plasmids to toxins, SCFA. I'll get more into that later, but that stands for short chain fatty acids. And we'll talk more about that in human microbe interactions. But basically, it's just cell membrane material or the outer membrane of gram negatives. So it's phospholipids, basically, that form a vesicle. And they get released and kind of float away from the cell. And if there's another nearby cell, then perhaps that vesicle can interact with that nearby cell. And so again, a way for the whatever's inside the vesicle can get transferred over to someone close by, and then that can get absorbed by that microbe and deliver the whatever's inside. And so again, it's a way to get chemicals and information, form sensing signals they've even thought might be part of this. But basically, it's a way to get these vesicles kind of moving away from the cell and interacting with other neighbors. They've even speculated that a microbe might release vesicles to fake out, so to speak, phages that are trying to attack, which is pretty cool. If that holds to be true, that's a pretty good strategy where if you can sense that there's plenty of phages around you wanting to attack you, you can, essentially release these vesicles and like, go attack that, leave me alone. And so the phages will go to the vesicle thinking it's a microbe and it's just a vesicle. And so they've shown that that can happen. So that's a really, really interesting idea of one way that microbes, bacteria can try to defend themselves by creating a decoy, essentially. So this is, they've known about vesicles like in humans. It's actually used in to deliver medicines, for example, into humans and other animals. And this liposomes essentially is a vesicle. You might have heard of liposome treatment. You can put some kind of drug inside of it and then get it to fuse with the membranes of the cells and deliver whatever inside. So this isn't brand new, but the fact that microbes are doing this too and using it as a way to get things delivered from one cell to the next in the neighborhood is pretty interesting. So here is a paper that was published. In this case, they were, talking about very unrelated organisms capable of interacting through this process, and including the short chain fatty acid idea, talk about that, and form sensing, peptides perhaps. And so this particular paper was actually looking at the process of it and then thinking about how to use that to deliver. I think they were looking at herbal plants and some of the chemicals that for medicinal purposes. So, but they're looking into it more to see how we could capitalize on this and use it in medicine. But again, this is just a visual trying to show this idea of how it pinches off. And they have such interesting words, blebbing. I don't know if that's used in other vesicle ideas, but they call it blebbing. There's explosive cell lysis. There's a possibility, here they call it bubbing. I don't know why they come up with these words, but kind of cute. So you don't need to know those. I just thought it was kind of funny. And it's a nice visual showing, you know, this idea that it's a bilayer membrane that's, you can have certain things captured inside of it, so. And then this was taken from another paper where they're trying to show like, what could this be doing for the, you know, the microbe that might be producing it, and how could it work ecologically? And so they have several ideas. They're looking at how it could maybe help the pathogen be more pathogenic, or you could use it as a decoy, like I said, for phages, toxin delivery. I mean, there's kind of lots of options to think about. Here they're looking at vesicle cargo binding nutrients and delivering it. Here you have a toxin that's kind of attacking the whole cell that then releases the nutrients that then can get delivered back to the producing cell. So lots of ideas, biofilm formation and dispersal, they're looking at it. So extracellular vesicles, much more common than they thought. They thought it was kind of a one-off, but the more they look, the more they find it. So this is another mechanism to think about for communication. But that's all I'm going to say about that. There's not a ton of research on that yet. So we're still in the early stages. All right, so let's do a Google Forms. Whoops. Okay, so now into the next slide set. So the fungi. So we're going to get into this tomorrow in lab, and basically you're going to be trying to learn the different morphologies of fungi visually, both macroscopically. We've grown some up on plates. And did some of you take some plates home to try to capture? Were you able to capture some fungi? Yeah, I haven't looked at mine yet. I'll have to go. I have them in the incubator. Ooh, yeah, nice.
I don't know what kind it is, but I want to.
Yeah, you'll definitely, I wonder if we have dissecting scopes in that lab. That's definitely a fun one. And some bacteria on it too. But yeah, the white fluffy fungus. So here you're looking at some different growth forms, and this is not white fluffy, it's white, but it's not fluffy. This is more yeast-like. So if you grow bread yeast, which you'll look at tomorrow, you'll see plates that look like that. And so those are unicellular type fungi. They grow in a unicellular manner, which you're seeing here in the lower left image. And so you probably won't need to use oil immersion really if you don't want to, because even though they're unicellular, they're so big compared to bacteria, it's going to be easy to find once you get your microscope in focus. And then the top images are showing more of the mycelial or hyphal growth form. And so those are the long filamentous type growth forms. And so there's different kinds of these. We'll talk a little bit more about that. And then if you have a bunch of mycelia all coming together in a very tissue-like organization, when they want to go through sexual reproduction, then you start talking about some kind of fruiting body. And so a lot of you are very familiar with seeing those when you're out hiking around. So we're into the kingdom fungi. So we're jumping out of the prokaryotic world and into the eukaryotic world. So they're eukaryotic like, we are. And they are in their own kingdom. And none have been discovered that can do photosynthesis. And all of their cell walls, they do have cell walls, but it's not peptidoglycan type thing. It's chitin, which is what you see for insects, the exoskeleton of insects. And so their mechanism for going into, like, how do they disperse? How do they spread? Very often their strategy is to do spore formation, because that can get, if the spores get up into the air current, it can waft a fair distance. Or if an insect crawling along and gets covered in spores, then the spores get carried off that way, for example. And the spores can be both sexual and asexual. I'll touch on that. And then we have the unicellular type fungi, which we often refer to as yeast. And then just be aware that under the right conditions, certain yeasts can switch their morphology. They can be unicellular, but given the right triggers, they can switch into hyphal growth forms. And so far, the ones that are most famous that can do this are human pathogens. And so we'll look at a couple of those ideas. And then filamentous is the other growth form. So unicellular or filamentous are the two growth forms, two morphologies. And if they are filamentous, then we tend to say the word mold. We say, oh, look at the mold. And so here on the lower left image, that's a mold. Close up of it is being shown in the middle here, where you see all this hyphal growth, and then the dark splotches are where the spores are formed. And then here, this very top image is a beautiful image of a hyphal, a hyphae. And then at the end of the tip of that, you see all these spores getting produced. This is a very classic image of Penicillium, that genus that gives us penicillin. They call this the paintbrush form of spore formation. And it looks like a paintbrush, so they named it, they give it that nickname. So when you look at fungi, there's certain things you can say they all do, which is they excrete out enzymes, very similar to how bacteria live their life. They have to excrete out enzymes because they're so small. Those enzymes break down the complicated polysaccharides or whatever, and then they absorb the broken down nutrients. So that's very similar to what we see with bacteria. If they have a hyphal growth form, it's usually, if you look along the whole length of the hyphae under the microscope, you'll see many nuclei showing up. It's not just one nucleus for this big long strand, there'll be many nuclei. And if you are talking about like one of the filaments or a handful, we tend to say the word hyphae. That's plural. Okay, if you want singular, you'd say hyphal or hypha. Say hypha or hyphal, A-L on the end. Hyphae is plural. And then if you have a bunch of hyphae all together, then we start using the word mycelium. Okay. So just some jargon that has to be learned on that. And then they all seem to have chitin, no matter what type of fungi they are, their cell walls have chitin. And we're not going to get into that like we did with peptidoglycan, but again, it's similar to what we see with insect exoskeleton. And they have done studies where, per gram of chitin or whatever, it has the tensile strength that's actually stronger than steel. So incredibly effective for creating a nice cell wall so that they have some structural integrity and protection. And then if you look at the cell membrane of all the fungi so far, they have what's called ergosterol, which is their version of cholesterol. It's not cholesterol, but it is close to that. And they don't see that in animals or plants. Like we have cholesterol, plants don't have ergosterol. Plants have other things besides cholesterol. So here again, looking at the terminology where you're thinking about one or just a little bit, like hypha is just one. You look at a couple, you might say hyphae, and then a whole bunch, you say, oh, mycelium. And then this is just, if you're curious about chitin chemical structure, that's what it looks like. So then the idea is, like when you look at the, where did it go? So when you look at this image in the lower left, you can see how symmetrical it is and how it's, very nicely formed. And so what they did to get this plate to develop is they put a chunk of mycelium in the middle of the plate and then let it incubate. And what you have to imagine are the hyphae that were right in the middle there. They start to grow out in like a radial pattern from the tip of the hypha. All right, so it's a tip growth mechanism. So let me go back. So it keeps expanding at the tip. And when they look at it, it is a combination of turgor pressure, so water pressure of all the water in the cytoplasm. of the hyphae is pressing against the cell membrane, pressing against the cell wall. And that's creating a nice force to like push and expand. And then they actually use membrane material from the endoplasmic reticulum. So they'll grab some of that and fill in the growing tip as they keep expanding. So it's tip growth. It doesn't start like at the base of the hypha, it's always at the tip. And so they keep exploring, and when you grow a fungus on that plate like that, that's how come it does that nice radial symmetrical growth pattern. It's the hyphal tips all expanding out. So here is an kind of a mock-up of this idea where they're trying to show, you know, they got on here, we're not going to get into the details, but I like this because they're showing the idea that yes, Turner pressure is involved and that you have vesicles coming off the endoplasmic reticulum. So that's basically what I want you to know. We're not going to get into step by step by step. So if you inoculate a Petri plate in the center like that and let it incubate, it'll form this very symmetrical growth pattern. And so what's happening is the freshest or most vigorous hyphae is what's on the edge of this circular growth. The older hyphae is more toward the dead center. And so if someone gives you that plate and says, hey, subculture this and get some more of this growing up, would you? You don't want to take the center. You always want to take edge, all right? And so I often will take, like with the naked eye, I can see where the edge is, and I'll take some of that, but I'll go out a little bit into what looks like bare auger, but it's not. There's hyphae there too. You just can't see it yet with the naked eye. And that's the most vigorous, healthiest hyphae that you should capture if you want your subculture to work and grow well. Okay. What about the yeast, the unicellular? This is a really nice image showing a yeast cell that has had many babies. So this budding going on, a bud stock. This is, and then it's actively budding here. And so with yeast, that's how they tend to reproduce. And so Saccharomyces cerevisiae is bread yeast, or it's also the yeast that's used in the brewing industry. And it can do that over and over and over. And so you can get many scars on the so-called mother cell that can repeatedly bud like that and produce. And the budding cell is usually smaller, but it'll grow to its typical size. It just starts out a little small. It's like little baby yeast that will grow up. Okay. And so you can buy that off the shelf as baker's or brewer's yeast. They use it in the brewing industry too. Many different species and strains of Saccharomyces are used in the alcohol industry. Candida albicans is very commonly found associating with humans and other animals. It can be pathogenic to humans, but often not, and can just be hanging out with you. But it is opportunistic. If the conditions are right, it can gain too much of a foothold and start to cause problems, too much growth. So thrush in the mouth, babies get that commonly. And so that kind of idea. And then this is the one that can be also dimorphic. It can be unicellular, but then it can shift into hyphal growth form. And so again, often pathogens display that kind of ability. And sometimes they think that it has something to do with their ability to get access to nutrients, why they make that switch. There's some nice pictures in the lab PowerPoint that get at that idea. So here's a mock-up of the yeast type fungi and showing kind of a complicated life cycle. I'm not going to get into the life cycles in any detail. Different classes of fungi do it differently. That's how we classify them. We figure out how they have sex. So that's part of the job, is you try to get them into the lab and create situations where they have sex so you can figure out where to put them in the classification scheme. So here, basically what I wanted to show on this is that yeast are pretty versatile. They have the ability to form and just like clone themselves with the bud scar idea or the budding idea, or they can go through fancier sexual reproduction. So in fungi, we don't talk about male, female. We don't say that. If for sexual reproduction, there's no male versus female fungi, it's plus minus. We say they're plus mating type, minus mating type. And the way we talk about it is that a plus and a minus strain have to merge in order for mating to be successful. If you have two pluses, they're not compatible. Two minuses, they're not compatible. So opposites attract, even in the fungal world. And we have that. So I'll show you some other pictures of that. But basically, yeasts are kind of funky. They can do asexual reproduction and then sexual through this idea called alternation of generations, which we talk a lot about in the plant world. That's what plants do. So it's kind of interesting that they've adopted this ability. So, but again, I'm not going to get into the details of this. We even call it schmooze. I don't know. How would you pronounce that? S-H-M-O-O-S, schmooze? I've never heard that before. Again, really interesting term, funny looking term. So unicellular yeasts, especially Saccharomyces, I would like you to know Saccharomyces cerevisiae. It is famous. It is such a famous species because, and as this is showing, it's like a workhorse. It's like the guinea pig of the fungal world in terms of how we use it for research. It is used so extensively, and it has given so many breakthroughs in human medicine and what we can use yeast for, because they're eukaryotic, we're eukaryotic. So you can see here some ideas of why they're using yeast in the research world and what they're studying, but it is really such an amazing model organism that's being used in the fungal world. So really interesting. So here is just a table showing you kind of a compare and contrast of if you have a mold type versus a yeast type, what are we talking about? So septate, that's a new term, septate and aseptate. So septate means that when you look at the hyphae under the microscope, you can see cross walls along the length of the hyphae. And they're not solid. So there's usually a little pore or tunnel right in the center. So it does allow for cytoplasm to stream along the length and some small chemicals, and not nuclei generally. It's too big for that and other organelles, but there is a pore. And then aseptate just means it's one big long hyphae. It's all open. Okay, so that's actually something we look at if we're trying to identify a fungus. We do look at that under the microscope. And then just remember the mycelium term. If you have a bunch of hyphae all together, you call it a mycelium. Here with yeast, we said they're unicellular. They can be dimorphic. Budding is often done. They're how they make more. Pseudohyphae, I haven't shown you that. So that is not true hyphae. But when you look at it under the microscope, you might at first think it's hyphae, but basically all it is yeast cells kind of stuck end to end to end into a chain. That's really all it is. But if you're not looking carefully, you might go, oh, look at the hyphae, and it's not. So they call it pseudohyphae. And so Saccharomyces in Canada are very, very famous yeast type fungi. So yes.
If the yeast cell is dimorphic, does it create the regular hyphae or the pseudohyphae?
Yes, good question. So if it's dimorphic, they consider that true hyphae. It looks exactly like hyphae. Yeah. So the pseudohyphae is just essentially a bunch of yeast cells kind of stuck end to end, kind of like a pearl necklace. Kind of like that idea. Okay. Pictures. So here's the pseudohyphae idea where you can get the yeast cells. Sometimes the yeast cells elongate like that. It's kind of funky. So here's non-septate and septate. And then here under the microscope again, you're seeing that beautiful paintbrush idea where they form spores. And then this is yeast. You'll look at this under the microscope. You'll make some fresh slides and do some staining with that to look at them. Yes. Yeah. So if you see long filaments of fungal growth, each filament is a hypha. And then if you see like 5 or 6 of them, then we tend to say hyphae, the plural. And then if you see a whole mass of them, like just thousands of them, then we start using the word mycelium. So it's more a quantity thing of how they switch. So, but mycelium is just a whole bunch of hyphae that's clustered together.
Are those considered cellular still or not?
So they're multinucleate. Splitting hairs, but that's, you know, because if you look along the whole length of the hyphae, you'll see even whether it's septate or aseptate, you'll see many nuclei floating around. So, yes.
For the pores, are they good to be visible like by a human eye, or is it only like the ones that the
Yeah, I don't think our microscopes could pick up the pore. I don't know.
like I was thinking when it says crosswalk in that, right? I had some slime mold, I think it was in mine, and like it was visible, like it almost looked like skin or something.
Yeah.
But that's not a pore.
Well, so you can see the cross walls under the microscope because that's often how we help identify. Let me see if I can. I think in lab there's some nice photos in there. Huh. Well, I only have the mock-up of it, but basically when you look under the microscope, you can see those cross walls. You see that? And then, but like right in the middle of the cross wall will be like a little tunnel, and you can't pick that out, even with oil immersion.
And that's the pore.
Yeah, that's a little pore that allows for cytoplasm to go through the whole length, because you have to imagine that this whole length has to be fed. I mean, it all has to get access to nutrients, you know, and there's flow of cytoplasm along the whole length, you know, back and forth. Okay, so you're gonna see in lab that we're gonna focus in on mainly 3 different classes in the lab, and I'm gonna add a 4th in lecture. So when you look at the fungi, you know, in total and think about all the fungi out there, I think we've all seen fruiting bodies and we've probably eaten some too, because many are edible. They're fungi that are very symbiotic with other organisms. So lichens, if you've learned about lichens, they're part fungi. It's fungi and some kind of either cyanobacteria or something like that. Or an algae, something like that. So they're part of that structure. Amazing. Lichens are amazing symbiotic organism. They are in a mycorrhizal relationship with plants. So we'll talk about that a little bit. And of course, there's pathogens. That's probably what you're most familiar with when you think about fungals get a bad rap. They're often demonized out in the, you know, non-science world. But there's far more fungi out there that do very good things for us. And we're learning that more and more. So the classes I want you to know in lecture are 3 of what you're going to learn in lab. And then this last one, it will be not in lab, but I'm going to ask you to know it in lecture. So zygomycetes, ascomycetes, basidiomycetes, and then a relatively new one, the glomerulomycetes. So these are different classes. There's more. Those are the only ones I'm going to focus on in this class. And so, you know, if you are, if you are given a fungus and someone says, what is this? Then, you know, what you try to do is narrow it down to class. That's one of the first things you try to do. And the way you do that is you look at it under the microscope. You look for septate, hyphae, aseptate. Is it, you know, is it mold versus yeast? What's going on? And then you look at their spore production and the way they form their spores and how they kind of design it is very telling. That can help you classify. And then the way they go through sex. So you try to make them have sex in the lab. Like you try to create a situation where that will happen. And sometimes it doesn't happen. Sometimes we can't figure it out. But basically you try to get them going and get them to have sexual reproduction, because how they do that and then how the spores get produced through that is very telling and can classify them into these different classes. So sometimes when you're like given this fungus, you have no idea, and you're like, well, okay, I'll bring them into the lab and I'll see if I can have them go through sex. Some, we just can't get them to do it. And that's sometimes because we can't figure it out. We just haven't met their requirements. And maybe sometimes it's because they just don't have a sexual cycle. They just do everything asexually. So the fancy term for it, we have no idea. We call it mitosporic fungi. So that's a class or a group, like an umbrella term of, we can't figure this fungus out. Maybe it doesn't have sex, or we don't know what it does, if it does, because we can't get it to do it. So it used to be another class. They used to put fungi that we didn't know if they had sex. We put them into class Deuteromycetes. That was the old term. But sometimes it's called imperfect fungi. I'm getting my phrase mixed up because there's a company named Fungi Perfecti. Paul Stamets has a company. Paul Stamets is one of the fungal gurus out in the world of fungal research. And he has a company called Fungi Perfecti, but the imperfect fungi, that's what we used to call, but now it's mitosporic fungi. But anyways, oh, there it is, imperfect fungi. And so that used to be the old class Deuteromyces. So you'll probably see that somewhere in some writing or publication of some sort. Just be aware that that's kind of phased out. And they just say, oh, they're mitosporic. We have no idea how they have sex. Okay, so let's look a little bit at some defining features of the zygomycetes. So, oh, we're running out of time, aren't we? Is it 2:50 already? All right, we'll stop there and we'll look at some of the defining characteristics of these different classes. We'll do that on Friday, but have fun tomorrow in lab looking at fungi.